Welcome back to Biology 2225L, AMP2 Lab at CNM. This week's topic is the female reproductive system, and this is Chapter 28 in your current textbook. Now recall from our discussion last week that the female reproductive system is more complex than the male system because it has to carry out more functions. Not only does it have to produce the gametes, which of course the male reproductive system has to do as well, but it also has to provide the bulk of the nutrition to the zygote, because the male sperm cell only provides the nuclear material, recall. The female also has to house the developing fetus and nourish it, and it has to receive the sperm from the male during internal fertilization. So there are a variety of functions that have to be performed by the female reproductive system, and so we'll see its structure does lend itself to those functions. Also, the female system is more cyclic, especially in its hormonal control, and we will take a look at those cycles as well in this lab. Now, right up front, I want to comment on this particular PowerPoint. First of all, I've modified it quite a bit from the basic PowerPoint that you have access to, provided by the publisher. I've modified it in a couple of ways. First of all, I've removed a lot of slides that we're not going to discuss in the lab. Secondly, I have corrected some errors that are in the PowerPoint. There are a couple of places where there are some fairly glaring errors. So I've crossed those out and corrected those. And the sequence is, as is so often the case with this particular textbook, not the same sequence that we present the terms in our list of terms. I haven't corrected that because that would have been too much of an overhaul. But I will point out as we go through it where we're jumping around a bit in the sequence. I've also modified some of the slides because when it comes to terms like ovarian cycle, sexual cycle, reproductive cycle, menstrual cycle, different books utilize those terms slightly differently, and this book has a particular way of talking about each of those things that doesn't necessarily correlate perfectly with what we have in our list of terms. So I've tried to modify some of the slides later in the presentation to fit a little bit better with what we have in our list of terms. And then finally, I have added some slides to the presentation that I took from another book, specifically slides having to do with the fluctuating hormones that are associated with the sexual cycle, that I think will clarify better what's going on, better at least than the single slide that your publishers present in your presentation. So you will notice as we go through this, that there are some differences between what I'm presenting here in terms of sequencing slides, some corrections to errors, and so on. All right, so as you may recall again from our conversation last week, the two sexes pretty much begin the same in embryonic development for the first 8 to 10 weeks or so. They're essentially the same, and then due to the effect of androgens, male hormones, the default state of female begins to change into male of course, the male. In the female reproductive system, in the absence of those male androgens, the female will continue down the default developmental pathway of becoming a female. So we don't have any specific hormones that trigger female development. It is the default state. It's the absence of things like testosterone and another hormone called Mullerian inhibiting factor that cause the female to stay on the female developmental track. The genitalia of the female are generally split up into internal genitalia and external genitalia. In our list of terms, we present the external genitalia first, and then we move to the internal genitalia. This particular PowerPoint takes the opposite approach, and so we will be discussing the internal genitalia first, and then we will move on to the external genitalia after that. The internal genitalia consist of things like the ovaries, which are the primary sex organs themselves that produce both hormones, and the gametes, the ova or oocytes. The uterine tubes or fallopian tubes, which carry the egg from the ovary to the uterus itself, of course, which houses the developing embryo/fetus, and then, of course, the vagina, which is designed to receive the male organ for internal fertilization. The external genitalia, of course, can be seen from the outside, and they include the clitoris, which is analogous to the male penis and has lots of sensory neurons in it, and folds of tissue that enclose the various orifices associated with this part of the body, the labia majora and the labia minora. So again, the primary sex organs are the ovaries that produce the gametes themselves, and then all the other organs that we'll talk about are referred to as secondary sex organs. And here we see a sagittal section through the female reproductive system. We will come back to this as we discuss various structures along the way. And here we see an external view of some of these structures, and again, each of these structures we will discuss in order as we go through this PowerPoint. All right, so as I said, this particular PowerPoint chooses to begin with the internal organs, and it begins with the primary sex organs, the ovaries themselves. These are the gonads of the female, and as I said, they produce the egg cells and the sex hormones. They are located in the pelvic cavity of the female, and they are not free-floating, of course. They are attached through various sheets of connective tissue to the pelvic wall. The ovaries, like the testes, have a protective capsule called a tunica albuginea, and then internally they have a cortex and a medulla. The outer cortex is where the gametes develop. The inner medulla is made up mostly of connective tissues and arteries and veins, lymphatic vessels, and so on. The eggs that we're going to talk about develop in structures known as follicles, and a little bit later in this presentation we'll talk quite a bit about the different kinds of follicles and the different stages of follicular development that we can observe in the ovaries. The event that we call ovulation is the bursting of one of these follicles to release the actual egg cell. And it's technically incorrect to call it an egg cell, because what is ovulated is a cell that has not even completed meiosis yet. And so it's more correct to refer to it as an oocyte. But again, we'll come back to those details in just a bit. Here we see a histological view of an ovary. And again, what we see primarily here is the outer layer called the cortex, and then a inner layer known as a medulla. And this medulla, just this part here, is the medulla, and again, nerves, lymphatic vessels, blood vessels, etc. And so all the follicles containing developing oocytes are found out here in the cortex layer. The ovaries, like the fallopian tubes and uterus, are all held in place within the pelvic cavity through a variety of ligaments. Later, when we discuss the uterus, we'll revisit these ligaments. Right now, we're just going to mention really the two that are most involved in helping hold the ovaries in place, and these are the suspensory ligament and the ovarian ligament. The ovarian ligament extends from the side of it's a cord-like band of connective tissue that extends from the side of the uterus out to the ovary on each side. The suspensory ligament comes down from above. It comes down from the roof of the pelvic cavity and the side wall of the pelvic cavity, and comes down and anchors or connects to the ovary from the top and side of the ovary. And so literally it's what helps suspend the ovary out to the side away from the uterus. The suspensory ligament contains the main arteries, veins, nerves, and lymphatic vessels that will then invest the ovary itself. The ovary is also on its posterior side anchored to a very broad ligament, a broad sheet-like piece of connective tissue called the broad ligament, which is an important ligament that helps hold the uterus in place. The ovary is in front of that broad ligament, but does have a sheet on its backside that attaches it to that broad ligament. That sheet, by the way, is called the mesovarium. Then, as we said, the suspensory ligament is where you find most of the veins, lymphatic vessels, and nerves that serve the ovary. And so we go back to the structure of the ovary, and we're going to discuss exactly what's happening in these follicles later in the presentation. Right now, our goal is to simply be able to identify them. And so what we find is that in the cortex layer, at the very surface, we have many so-called primordial follicles. And by many, I mean potentially hundreds of thousands of primordial follicles, at least initially. That number reduces quite a bit as the individual goes from childhood into adulthood. At any rate, as part of the basic cycle, which we'll talk about much more detail in a little bit, primordial follicles turn into primary follicles. Primordial follicles can be distinguished by being surrounded by a simple squamous epithelium. By the time they develop into a primary follicle, the epithelium is changed to more of a simple cuboidal. From the primary follicle, it continues to enlarge into a secondary follicle. A secondary follicle generally has a thicker cuboidal epithelium around it, though the primary follicle can have more than a single layer as it starts to turn into a secondary follicle. The key feature of a secondary follicle that most books talk about is you begin to develop a little fluid-filled space known as an antrum. And so even though it's not shown here, the secondary follicle should contain a very small antrum. Your book talks about a tertiary follicle, which I think most books consider to be just a late secondary follicle, and we see that fluid-filled space starting to develop. At the very end of follicular development, we end with something called a vesicular or Graafian follicle. And a vesicular or Graafian follicle is a very characteristic shape. Most of its volume is the fluid-filled antrum, which has expanded now. And the oocyte, the so-called egg cell, is projecting or protruding into that antrum, and a very characteristic stalk-like structure. And so this is known as a vesicular or Graafian follicle. And this is what a mature follicle would look like right before ovulation. When ovulation occurs, what happens is the mature follicle will literally rupture to release the oocyte and some of its surrounding tissues. And the fluid, of course, of the antrum is released as well. After ovulation, the Graafian or vesicular follicle that ovulated becomes a structure known as the corpus luteum. The corpus luteum is a glandular structure. Luteal means yellow, and it literally is yellow in color due to the accumulation of fat, adipose tissue within it. But the glandular tissue of the corpus luteum becomes a very important source of hormones that are going to maintain pregnancy if the ovulated oocyte is fertilized. If the ovulated oocyte is not fertilized, then the corpus luteum, within a matter of days, will begin to degenerate and turn into essentially a mass of scar tissue known as the corpus albicans. Alba is the word for white, and again, because it's composed mostly of collagenous fibers at this point, it becomes a white body. So corpus albicans versus a yellow body, corpus luteum. So this is what we need to be able to recognize, at least in terms of how the follicles of the ovary change during the typical cycle. But again, we're going to come back and talk in much more detail about what's happening within each follicle during these phases, and specifically what the hormones of the corpus luteum are, etc. So more detail on these things to come. In terms of recognizing them under the scope, we have a variety of histological views for you coming up. So here we see the primordial follicles, the very first kind of follicle that you have in the ovaries. And again, you'll note that they are essentially an oocyte surrounded by a thin layer of simple squamous cells known as granulosa cells. Another view of the primordial follicles with their simple squamous granulosa cells surrounding them. There's a nice view of simple squamous. A primary follicle is a bit larger, so you can see it is expanded around the oocyte, the primary oocyte, and it has a lining of simple cuboidal or slightly stratified cuboidal epithelium as it gets closer to its conversion to a secondary oocyte or secondary follicle. So a single to just a couple layers of cuboidal cells is characteristic of a primary follicle. And here we see another view again of primary follicles with their granulosa cells, this time cuboidal and 1 to several layers thick. Now your book at this point, your PowerPoint at this point goes on to show a secondary follicle, but unfortunately what it shows is a vesicular or Graafian follicle, and it lists it as a secondary follicle. So that's one of the several mistakes that we have in the PowerPoint. So I've replaced that image with what I believe to be a more accurate view of what a secondary follicle looks like. And what you see here is, again, the oocyte is at the center, and you have a much thicker zone of granulosum cells. So clearly not just 1 to 2 or 3 layers thick, but multiple layers of cuboidal epithelium. Again, these are called granulosa cells, or the zona granulosa. There usually is a light-colored layer between the oocyte and the zona granulosa. This is known as the zona pellucida. Pellucida means sort of transparent, and so a little light transparent zone, zona pellucida. And these layers will persist throughout the follicular structure from here on out. Is there anything else distinctive about a secondary follicle other than the thickness of its granulosa cells? And the answer is yes. As I mentioned earlier, usually by the time you get to a secondary follicle, you begin to see the formation somewhere of a fluid-filled antrum. Usually, actually, what happens is you have several pockets of fluid that begin to pop up or arise in the secondary follicle, and eventually they will all fuse together to form one single fluid-filled space called an antrum. And then finally we get to the very distinctive vesicular or Graafian follicle. Again, I've renamed this because your book refers to this as a secondary follicle, and that's just incorrect. So remember, with a mature or Graafian or vesicular follicle, you've got a large C-shaped antrum with the oocyte projecting into the antrum on a stalk-like structure. Again, still surrounded by a zona granulosum, a pellucidum layer, right, a transparent layer. And there are some other terms that you could learn, but we're going to stick to the list of terms, and just all we have to know for this is that this is a Graafian or vesicular follicle. The corpus luteum is a highly folded structure. Again, post-ovulation, this is what a Graafian follicle turns into. And again, it's not yellow here because it's stained, but in real life it has a yellow appearance due to the accumulation of fat tissue within it. And then finally, if pregnancy doesn't ensue, or if pregnancy does ensue after 9 months and after delivery of the fetus, the corpus luteum will eventually degenerate into a massive whitish scar tissue known as the corpus albicans. All right, so now we're going to move on to the uterine tube. This is also known as the oviduct, and it's also known as the fallopian tube. It is a muscular canal about 10 centimeters long. 10 centimeters puts it at just under about 4 inches or so. Of course, we have 2 of them, a left one and a right one. And the job of this tube is to pick up the ovulated oocyte from the ovary and transport it towards the uterus. So it is a muscular tube with peristaltic waves of smooth muscle contraction that help propel the egg towards the uterus. It is also lined with a ciliated columnar epithelium. And the cilia help create, of course, a flow of fluid, which also helps draw in and transport the ovulated oocyte towards the uterus. Now, it's possible for an ovulated oocyte to not get drawn up into the uterine tube. It's possible that it might escape and wander around the pelvic cavity. If that does happen, it's possible that it can implant on any other tissues, the wall of the pelvis, the surface of the urinary bladder, and so on. It's also possible that sperm cells might be able to swim through this tube and into the pelvic cavity, and possibly, though it's rare, possibly even fertilize an oocyte that is implanted in an incorrect position. This could result in a rare kind of ectopic pregnancy where you get initial development of a zygote somewhere other than the uterus. The most common type of ectopic pregnancy is where the oocyte gets stuck in the uterine tube, gets fertilized by a sperm cell, and begins its development within the uterine tube. This is known as a tubal pregnancy. None of these types of ectopic pregnancies, of course, will be successful, and they always result in spontaneous abortion. One other interesting thing to note about the uterine tube in terms of timing, if an oocyte is going to be fertilized by a sperm cell successfully, and then successfully implant on the wall of the uterus afterwards, the fertilization event has to happen while the oocyte is traveling through the uterine tube. If it happens before that, right at the point of ovulation out of the ovary, then the oocyte will arrive at the uterus too late and won't be able to implant successfully. If the fertilization event happens after the oocyte has moved through the uterine tube and has made it into the uterus, then the zygote will implant too late on the wall of the uterus. Too late. And also will result in unsuccessful. So I can't remember if I said that right. So again, if it happens out at the ovary, the implantation will be too early. If it happens out at the uterus itself, the implantation will be too late. All right, so we're back to this diagram again that we were looking at, and so we've talked about some of these ligaments and the ovary itself, and now we want to take a look at the uterine tube. Again, it's a hollow muscular tube extending from essentially the top side of the uterus itself. This is the uterus, and it extends out, curves over the top of the ovary, and then has a funnel-shaped region that with finger-like projections called fimbriae, and this funnel-shaped region, which is known as the infundibulum of the uterine tube, and these fimbriae are designed to help funnel or collect an oocyte that might ovulate from anywhere on this ovary. So there is a current and a suction that created by the peristaltic waves of contraction in the cilia that line the uterine tube that should draw the oocyte up into the uterine tube. But of course, you can see it's possible that an oocyte might be ovulated and wander away from the uterine tube, and it could implant anywhere in the pelvic cavity. But under normal circumstances, it gets drawn up into the infundibulum and then will move towards the uterus. In the meantime, of course, sperm cells working against that female obstacle course are swimming against the current and the muscle contractions, and again trying to find their way into the uterine tube where successful fertilization should happen somewhere in here, as we said. Here you see the inner lining of the uterine tube and how very irregular it is, how highly folded it is. And here we see a close-up view of the columnar epithelium. Generally speaking, it's a simple columnar epithelium, and as we said, of course, it is ciliated to help create that current. And here we see an electron micrograph of the columnar cells with their cilia that line the uterine tube. As we said, the uterine tube has fimbriae, little projections, finger-like projections at the tip that help gather up the ovulated oocyte. From here, we move on to the uterus. The uterus, of course, is a very thick muscular sac or chamber that will lead to the vaginal canal or vagina. Usually the uterus sits on top of the bladder. It tilts forward and so sits on top of the bladder. This is one reason why during pregnancy it is difficult to hold as much urine as normal, because of the uterus sitting on top of the bladder. As the fetus grows, expands, and develops, of course that compresses the bladder, which means you can't hold as much urine in it. The function of the uterus, of course, is to house the fetus and provide nutrition, ultimately through a structure known as the placenta. Its second function, of course, is when a pregnancy comes to term. Of course, the uterus has to be able to expel the fetus during delivery, and that really is why it's such a thick muscular chamber. The muscle part is involved in labor. The uterus has a variety of portions to it. It has a dome-shaped top, much like the stomach has a dome-shaped top, and that is referred to again as the fundus of the cervix. Most of the cervix is the body or corpus of the cervix. That's the more vertical central portion of it. And then finally, the bottom of the uterus, I may have said cervix, the body of the uterus is the more vertical central part. And then the bottom part of the uterus is referred to as the cervix, and the cervix is a very muscular tube-like structure that forms the inferior portion of the uterus and is, of course, the boundary between the rest of the uterus and the vagina. So here we see a sagittal section through a cadaver. Again, the uterus, which, as we said, this is anterior on the right and posterior on the left. We said the uterus tilts forward and sits essentially on top of the bladder. And here we see an empty bladder just underneath the uterus. The fundus, again, the dome-shaped top. The more vertical region is the body. They forgot to unhighlight the fundus, so it should just be this region here. And then the cervix is the very muscular bottom part of the uterus. As we said, the uterine tubes emanate from the upper two corners of the uterus, and in a non-pregnant uterus, you do have a space inside the uterus, but it's completely collapsed. So it's more correct to call the inside of the uterus a potential space in non-pregnant women, because the inner walls are essentially collapsed and touching each other. But of course, if you spread it out, such as during pregnancy, that becomes a real space. The cervix has an opening in it called the cervical canal, and of course this is how sperm can get from the vagina through the cervix up into the uterus. The internal os is what we call the top part of the cervical canal. So the internal os is the opening that's on the side of the uterus. The external os is the opening on the side of the vagina. And of course the cervical canal and the cervix is what's palpated during pregnancy, feeling to see how much it's been dilated, and that's usually a good indicator of how far along the pregnancy has progressed, because the cervical canal has to expand or dilate as part of labor and delivery. There are glands lining the walls of the cervix, and these glands secrete primarily mucus. The mucus helps slow down the spread of microbes from the vagina up into the uterus. These secretions can also drip down from the cervix and help to lubricate the vagina during intercourse. And so here again we see a diagram of the system. Of course, the uterus here with its dome-shaped top called the fundus, the body, the more vertical portion here, and then the cervix down here. This opening on the, again, uterus side is called the internal os. The opening on the vaginal side is called the external os. You'll also notice that the cervix kind of hangs down into the vaginal canal, which is down here, and that there are portions of the vagina that seem to go up along the sides of the cervix. These are referred to as the fornices of the vagina, the lateral fornices that go up each side of the cervix. Now, the wall of the uterus is broken down essentially into 3 layers. On the outside, we have a connective tissue capsule known as the perimetrium. This is not on our list of terms, but you may want to add it, since to have a complete discussion of the wall, we really should include it. So the outer fibrous capsule is the perimetrium. The majority of the uterine wall is called myometrium, and that consists of smooth muscle tissue. And then the inner lining of the uterus is called the endometrium, and the endometrium is going to be the most important part that's involved in nourishing and implanting a zygote, and then nourishing the developing embryo / fetus. The myometrium, of course, is most important during labor and delivery. So again, perimetrium, the outer layer, the myometrium, the muscular layer, which again constitutes most of the uterine wall and is composed primarily of smooth muscle. And the arrangement of the muscle fibers is such that when they contract, you get a wave of contraction downward from the fundus, which makes sense if you're trying to expel a fetus, and it tends to have a twisting or spiraling motion as well. The endometrium is the inner lining again, or the mucosal lining of the uterine wall, and again it's involved in implantation and nutrition of the developing embryo and fetus. It is lined with a simple columnar epithelium, and we have many tubular glands in the endometrium. The function of these endometrial tubular glands is still being researched. Most people accept that they play a role in implantation, providing the proper environment for a fertilized egg, a zygote, to implant, and then they also play some role in secreting nutrients for the developing embryo, especially before the placenta develops. There's some evidence also that these tubular glands might contribute to some of the structures associated with the developing embryo, various membranes like the chorion and the amnion, and so on. The endometrium actually is broken up into 2 layers, a functional layer and a basal layer. The functional layer, of course, is the part of the endometrium that does most of the work of accepting a zygote and then developing to nourish it during pregnancy. This is the part that is sloughed off on a 28-day cycle if pregnancy doesn't ensue following ovulation. The basal layer is the deeper layer of the endometrium, which is closer to the muscle, the myometrium, and this layer does not slough off. This is the layer that stays behind, and following menstruation will regenerate a new functional layer, again on a 28-day cycle. And we're going to talk quite a bit about this 28-day cycle in just a bit. And so here we see a histological view of the wall of the uterus. Down below here we have smooth muscle tissue, that's the myometrium. This, from this stained region up to the surface, that's all the endometrium. The basal layer of the endometrium that's not sloughed off is just this bottom layer right here. Everything from about this point up, this is all the functional layer of the endometrium, and this is what gets sloughed off during menstruation again. The thickness of this functional layer will vary quite a bit over the course of the 28-day cycle, and so as we'll see, it gets progressively thicker and thicker as we get through that towards the end of the 28-day cycle. And then, of course, at the end, during menstruation, it's this functional layer that gets sloughed off. And so we go back to basically just the basal layer again, and we start to build up a new functional layer in a new cycle. A very close-up view of the surface of the uterus, again with primarily a simple columnar epithelium and these tubular structures, which are the endometrial glands. And yet another view of the wall of the uterus, specifically focused on the endometrium. And I think what's nice about this view is you can see that these endometrial glands are actually long tubular glands, somewhat reminiscent of eccrine sweat glands in the skin, just having a coiled secretory portion, and then with a tube, a duct leading to the surface. As we said, not only is the ovary supported by ligaments within the pelvic cavity, but so is the uterus, of course. And the uterus, being larger and, of course, much heavier, especially during pregnancy, has more ligaments that support it. We've already talked about the broad ligament, which is a sheet-like ligament that extends from the sides of the uterus to the walls of the pelvic cavity. Another ligament that's very important in supporting the uterus is the round ligament, one on each side. So two broad ligaments, two round ligaments. The round ligament arises from the front of the uterus, passes through the inguinal canal, and actually terminates way down underneath the labia in the external genitalia. And so it is by passing through the inguinal canal, it's similar to the gubernaculum, which includes the spermatic cord in the male. So this helps, again, hold the uterus in place, but it anchors in an unusual place closer to the outside of the body wall. There are some other ligaments that are smaller that are also helping to hold the uterus in place, but the two main ones for our purposes are broad ligament and round ligament. Of course, as the uterus expands during pregnancy, these ligaments, of course, get stressed and stretched out quite a bit, and so that is the source of the so-called familiar ligament pain associated with pregnancy. And here we see some diagrams of these ligaments. The broad ligament, again, would be a sheet extending from the side of the uterus. This is the uterus looking down from above. This is the fundus, or top of the uterus. Here we see the fallopian or uterine tubes out to the side. There's an ovary right there, and an ovary on each side, and the fimbriae or finger-like projections. This is looking, again, from above down into the pelvic cavity. This is anterior down here, so this is the bladder. And again, the uterus kind of tilts forward, and as it expands during pregnancy, grows over on top of the bladder. The round ligaments are difficult to see, but the round ligaments, again, come from essentially the front side of the uterus. And again, they're going to pass through the inguinal canal and anchor down close to the external genitalia. Another view of the same thing. Again, just to review all the ligaments: the ovarian ligament leading from the side of the uterus out to the ovary, the suspensory ligament coming down from the top of the pelvic cavity and holding the ovaries out to the side of the uterus, the broad sheet-like broad ligament helping anchor the uterus in place against the walls of the pelvic cavity, and then the very cord-like round ligament right here running from the front side of the uterus towards the through the inguinal canal. And here we see again a cadaver uterus dissected out with the suspensory ligament out to the sides, the ovarian ligament running out to the ovary, little almond-shaped ovary there with the ovarian ligament. The broad ligament has been removed from this view so that you can see the round ligament that's going to run in again to the inguinal canal. The uterus, of course, does need a good blood supply to maintain pregnancy, especially, and provide nutrients, at least initially, until the placenta develops. So we don't want to say much about this. We don't have any terms on our list of terms. I just wanted to remind you that, of course, the blood supply is important. In terms of blood vessels that we've learned, remember we do have gonadal arteries and veins that serve the ovaries themselves. The blood vessels that serve the uterus and the vagina are actually blood vessels that branch off the internal iliac artery. So no surprise there, because remember we said the internal iliac artery provides blood to most of the pelvic organs, which of course the uterus and vaginal canal qualify. All right, speaking of the vaginal canal, again, your book chooses to take an inside-out approach, so we're moving from the internal organs out towards the external genitalia. So the vagina is also known as the birth canal, and of course serves two functions. Not only does it receive the penis during intercourse and is the initial receptacle for sperm delivered into the female reproductive tract, but it is also the canal through which the fetus has to pass during delivery. Again, it's about 8 to 10 centimeters, so again, just a little bit less than 4 inches long on average, and it is another muscular tube, and again, the muscle is smooth muscle tissue. And so, as we said, it receives the penis, it functions as the birth canal. It is also, of course, how menstrual fluid at the end of each 28-day cycle, it's how menstrual fluid exits the body. The sloughed off endometrium exits the body during menstruation. The vagina technically has no glandular tissue in it, though there is a kind of leaking of serous fluid through its walls, ultimately coming from blood vessels. This is known as vaginal sweating. And so even though there are technically no glandular tissue, the leakage of fluid ultimately from blood plasma helps lubricate the vagina during intercourse and delivery. And remember, there are cervical glands in the cervix above the vagina, and mucus and serous fluid from the cervical glands can also leak down into the vagina during intercourse and delivery, again to help with lubrication. As I mentioned, you have fornices, singular is fornix, little blind-ended spaces of the vagina that sit on either side of the cervix. There are some folds of mucosal or inner tissue that form a structure across the vaginal opening called the hymen. Now, in most women, the hymen is not a complete sheet or not a complete membrane. In most cases, the hymen is a sheet-like structure that does have an opening at the center, almost a donut-shaped structure. And so this is what allows young women who have not had any kind of intercourse to be able to menstruate during their first period, even though they have an intact hymen. In some rare cases, the hymen is completely cutting off the vaginal opening. It's a complete membrane, and of course before menstruation can occur, that hymen has to be broken. But in most, again, just to reiterate, in most cases it is not a complete sheet, and so it does not have to be broken to allow for menstruation. The epithelium that lines the vagina is different depending on what phase of life you're in. In childhood, the epithelium that lines it is a simple cuboidal epithelium. But at puberty, in anticipation of friction associated with intercourse, the epithelium is transformed into a stratified squamous, non-keratinized stratified squamous, which is a more appropriate tissue given the friction that occurs during intercourse and during delivery or parturition of the fetus. There is a microbial flora that exists in the vagina that produce a typical acidic pH in the vagina. This is not pathological. This is desired because, again, remember, acidic pHs are inhospitable to sperm cells, and the female reproductive tract is, in a way, trying to do a sort of selection against sperm cells, ensuring that only the strongest sperm cells make it through the vagina and up into the uterus and onto the uterine tube for fertilization. Here we see a sagittal section again through a cadaver. Remember, this was the uterus up here, the urinary bladder, and so the vagina again sits essentially below the uterus and behind the bladder, and it is this muscular tube. Notice the fornices that extend up along the sides of the cervix at the top. Here we see a histological view of the epithelial lining of the vagina. This, of course, would be an adult, in which case it is non-keratinized, stratified squamous. At this point, we're ready now to discuss the external genitalia, which are the very first set of terms that we have in our list of terms. So essentially, we've been working up from the ovary and the ligaments up towards the top of our list of terms. The external genitalia are collectively referred to as the vulva, sometimes also referred to as the pudendum of the female body. The external genitalia essentially consists of three major structures that can be easily seen at the surface. The mons pubis is a layer of fat tissue that sits on top of the pubic symphysis, and this is the part of the system that it bears, of course, most of the pubic hair, and the fat represents a cushioning. It helps provide a cushioning function during intercourse. Then we have two folds of skin tissue known as the labia, and remember, labium is a Latin word meaning lip. So labia majora, the larger major lips, are the external pair, and then within the labia majora, lying within the labia majora, is another smaller pair of folds known as the labia minora. Again, there are two, one on each side. Each one is referred to as a labium. So this one would be singular would be labium majus, and singular for this would be labium minus. But since we usually refer to them in the pair as the pair, we usually use the term labia majora and labia minora. The labia majora, again, are larger, they're thicker, they do have adipose tissue within them as well. They do have pubic hair on them as well. But again, within them are the labia minora, which are much thinner, do not bear pubic hair, and are essentially within or medial to the labia majora. So these three things usually can be seen from the surface. Now, within or enclosed within the labia minora is a region known as the vestibule. So everything within the labia minora is referred to as the vestibule. The vestibule contains the openings for the reproductive and urinary system. So within the vestibule would be the urethral orifice, through which urine, of course, exits the body, and, of course, the vaginal orifice, the opening to the vagina. As the labia minora come together at the anterior end of the vestibule, they fuse together to form a hood-like structure known as the prepuce. This is analogous to the male foreskin, and so the labia tend to be the labia majora are analogous to the scrotum. The prepuce, the fusion of the labia minora anterior, form a prepuce, which is analogous to the male foreskin. And just like the male foreskin or prepuce, they cover a sensitive a structure that is capable of enlarging and engorging with blood. In the male, that was the glans penis. In the female, this is known as the clitoris. Like the glans penis, again, it is erectile, and like the glans penis, it has lots of sensory neuron fibers in it, so it's highly sensitive to stimulation, and this is part of the female sexual response during intercourse. So again, as we just said, the clitoris is the erectile portion and sensory portion of the vestibule, and it even has a portion like the penis known as the glans in the body, and so on. Of course, just smaller than the penis. In this general region of the vestibule, we also have things called vestibular glands. The vestibular glands. These vestibular glands have an older name, Bartholin's gland, but generally we call them vestibular glands. Now, this is a pair of glands that open into the vestibule, and again are also involved in lubrication. Lubrication mostly during intercourse. So here we see a view of some of these structures. Again, the mons pubis is the fatty tissue that sits on top of the symphysis pubis. The perineum is what we refer to this entire region. The labium majus is singular for these two larger folds of tissue, skin tissue, with, again, that do bear pubic hair. The labium minus or labia minora are folds of tissue within the labia majora. And of course, the vestibule is whatever would be within the labia minora, enclosed by the labia minora. And that would include the vaginal orifice and the urethral orifice, which is not highlighted here, which would sit in front of the, or anterior to the vaginal orifice. As we said, the clitoris is a mass of erectile tissue that sits anteriorly, and as the labia minora come together at the anterior, and they form a sheet of tissue called the prepuce that encloses the head or glands of the clitoris. And here we see this in diagrammatic fashion. Again, the mons pubis. Behind that, the folds of tissue called the labia majora. Individual would be labium majus. Within the labia majora would be the labia minora. Singular would be labium minus. Within the labia minora is a region known as the vestibule. And again, in the vestibule we have the orifices, the urethral orifice and the vaginal orifice behind it. And at the anterior end we have the erectile clitoris, and notice the labia minora come together and form a collar of skin around the head of the clitoris, or glands of the clitoris, known as the prepuce of the clitoris. All right, so it's not on our list of terms, but just a quick comment about puberty. Puberty begins at age 8 to 10 for most girls in the United States, and that doesn't necessarily mean menstruation begins. Menstruation can be delayed to as much as 13, 14 years old, but the beginning stages of puberty can begin as early as 8 years old in the United States. This, of course, is triggered by hormones, primarily gonadotropin-releasing hormone, which comes from the hypothalamus, will begin to stimulate the anterior pituitary to secrete follicle-stimulating hormone and luteinizing hormone, which we're familiar with from our discussion of the endocrine system. Follicle-stimulating hormone, as the name implies, will begin to stimulate the ovarian follicles to go through their typical developmental cycle. Remember, we talked about primordial follicles, which turn into primary follicles and secondary follicles and so on. And so it's FSH that stimulates or initiates those follicles in that developmental stage. As the follicles develop, they themselves then begin to release a variety of hormones, most notably estrogen and inhibin are the two that are released primarily by the developing follicles. Of course, remember, after ovulation, a follicle turns into a corpus luteum, and then we can get the production of progesterone, which is, as we've said, a hormone that helps maintain pregnancy. And of course, as the ovaries begin to secrete estrogen in response to this FSH, the estrogen then travels throughout the body and causes the feminizing effects that we associate with puberty, which is, of course, breast development, hypodermal fat deposition patterns, pubic hair growth, and so on. Okay, so we've described most of the anatomy of the system. The remainder of our discussion today is going to involve the physiology of egg production and the hormonal control of the so-called menstrual cycle and ovarian cycle. Now, there's a lot of confusion. I don't want to call it confusion. There's a lot of different ways of talking about this, and different books use different terminology in different ways. And so we've all heard of the menstrual cycle, and some of us have heard of an ovarian cycle. But your book has a very particular way of talking about these things, and this slide is a sort of a good overview to keep in mind as we move forward. First of all, there's two kinds of cycles that your authors like to talk about. A reproductive cycle is the cycle of events that occur from fertilization to delivery of a baby. So that is known as a reproductive cycle, and of course that's a 9-month cycle approximately. The sexual cycle, on the other hand, is a 28-day cycle that recurs pretty regularly, again, when pregnancy doesn't occur. So when the female is not pregnant, she has a very familiar 28-day cycle known as the sexual cycle. Now, this sexual cycle can be broken up into two related cycles, just depending on what you're choosing to focus on. If you're focusing on what's happening in the ovaries during this 28-day sexual cycle, then your authors like to call that the ovarian cycle. If you're choosing to focus on what's happening, what kind of changes occurring in the walls of the uterus, the endometrium specifically, then we refer to that cycle as the menstrual cycle. So it's really all the same thing: sexual cycle, ovarian cycle, menstrual cycle. It's just that our particular focus changes, whether we talk about it as the ovarian cycle or the menstrual cycle. Our list of terms doesn't do a great job of distinguishing that. And so in the ovarian cycle, we just talk about what's happening with the egg cells specifically, but we haven't given specific names to it. But we'll go ahead and follow the book's convention as we go through the ovarian cycle and the menstrual cycle. So before we talk about either one of those two cycles in and give them any specific phase names, let's just talk a little bit about oogenesis. Oogenesis simply means the production of an ovum or an egg cell in the female. And what we're going to find, and of course this is analogous to spermatogenesis in the seminiferous tubules of the male, what we're going to find is that oogenesis is very, very different from spermatogenesis in the male. And just as a reminder, remember, spermatogenesis begins with stem-like cells called spermatogonia. And those spermatogonia line the periphery of the seminiferous tubules, and they begin with a mitotic cell division that produces two daughter cells, a type A cell and a type B cell. One of those daughter cells is fated to undergo meiosis and become sperm cells. The other daughter cell of that mitotic division remains at the edge of the seminiferous tubule and continues on essentially as a spermatogonium or stem cell. And so, as we said last week, males never really run out of sperm cells, even into their 70s, because they always have one of the two daughter cells of mitotic cell division remaining behind as essentially a functional spermatogonium. In females, the situation is completely different. We're still producing haploid gametes by means of meiosis, but the way in which it's done, when meiosis begins, where meiosis begins, how many cells are doing meiosis at the same time, all those things are very, very different. And of course, instead of a constant production of sperm cells like we see in the male, essentially, because of some cyclic changes that occur in hormones and the cycle of ovarian development itself, you generally only have a single ovum available for fertilization every 28 days. So again, that's very different. Males producing functional sperm cells by the millions every day, a female ovulating one receptive ovum every 28 days. So clearly very different kinds of gamete formation in the two sexes. So let's take a close look at what's happening during oogenesis. So remember that the ovary develops up by the kidneys during early embryological development. And during this early embryological development, we get stem cells or germ cells forming the initial cells, which are called oogonia. These are analogous to spermatogonia in the male. But again, this is happening during embryonic development. This isn't happening at puberty. This is happening before the individual is even born, early in their embryonic development. And in a typical female, you get about 6 to 7 million oogonia produced in the two ovaries while the individual is still an embryo. Those oogonia, those millions of oogonia, will enter the first meiotic division, all of them. Notice a couple of things here. First of all, they all enter meiosis completely. So there is no mitotic division before meiosis that's always going to leave some functional cells behind. All of the oogonia will enter meiosis. And so this is it. For the rest of the female's life, there are going to be no more oogonia available to make oocytes. So the number is set, again, early in embryological development before the individual is even born. And the second thing to notice is that, again, how early this is, that it's happening before the individual is born. And I said they enter meiosis, but notice it didn't say that they complete meiosis. They actually start meiosis and tend to get arrested in somewhere between prophase 1 and metaphase 1. So they don't complete the meiotic division, they just begin it very early in the development of the individual. Now, what happens to these cells? Well, first of all, once they've begun meiosis, they transform from what we call oogonia now into what we call primary oocytes. So a primary oocyte is a cell that has begun the first meiotic division. In our list of terms, we have just the term oocytes, but you probably want to be more specific. So under oocytes, you want to list primary oocyte and secondary oocyte. And right now, we're just talking about the primary oocyte. So it has begun the first meiotic division, but it gets arrested, it gets stuck somewhere in late prophase, early metaphase of that first meiotic division. Now, again, millions of them. We have millions of these oogonia that convert to primary oocytes. By the time an individual girl is born, most of those millions have degenerated. Most of them are gone. Because if you think about it, a woman does not need millions of egg cells in her lifetime. She only has, if you think about it, a female only has a reproductive life of about 40 years, and that's being generous. Let's say she becomes fertile at age 10 and goes through menopause at age 50. That's probably a good estimate of the reproductive life of a female. If she ovulates once every 28 days or so during that 40 years, essentially you're only going to ovulate about 480 functional egg cells, 480 functional oocytes. So there's no way that a woman needs millions of these oogonia. She would never use them during her reproductive life. At the max, she would only use about 480 to 500 of them. And of course, if she gets pregnant at any time during that 40-year span, that's going to reduce the number she would need, because during the 9 months of pregnancy, she won't ovulate. And so with multiple pregnancies, she'll need even less than that. So these millions of oogonia degenerate. Most of them degenerate primarily because they'll never be needed. All right, so by puberty, how many are left of these primary oocytes that have entered the first stage of meiosis? About 200,000 remain across both ovaries. And again, more than enough that she will need throughout her lifetime. And again, notice the big difference here. Males produce millions of sperm every few days. Females have a lifetime supply of gametes of only about 200,000, of which they're only likely to use about 480 or so. So big differences in the numbers of gametes produced. The other thing to notice is that by puberty, when the sexual cycle begins, all of these oocytes, all of these 200,000 oocytes in the ovaries have been stuck in meiosis since the girl was an embryo. So if her sexual cycle begins at, say, age 12, those cells, those primary oocytes, have been stuck in meiosis for over 12 years. So that's a pretty amazing thing to think about, because if you think about when you're in prophase 1 or metaphase 1, remember, homologous chromosomes are stuck together in the form of dyads. And so the chromatids, the arms of these chromosomes, have been stuck to each other, doing crossing over and recombination for over 12 years or more. And of course, as a woman ages and gets into her 20s and 30s, those cells will have been stuck together for 20 years, 30 years, 35 years, and so on. And this has some significance because, of course, to complete meiosis, you have to pull those tetrads apart into their dyads. The individual chromosomes have to separate. And as a woman gets older and older, it becomes more and more difficult to separate those chromosomes correctly. After all, if you're a 40-year-old woman, the egg you ovulate will have completed the first meiotic division after 40 years. And so the likelihood of them separating properly is reduced as the woman gets older and older and older. They've just been stuck together too long. And so pieces will break and extra pieces will travel to one cell and other cells will be missing pieces. It might occur that some chromosomes don't separate at all, and the entire tetrad, meaning 2 chromosomes, go to one daughter cell and no chromosome of that homologous pair goes to the other one. So this results in various kinds of birth defects, genetic birth defects. The most well-known one is trisomy 21 or Down syndrome, which is clearly correlated with age of the mother. Again, because of this odd situation where in oogenesis, the primary oocytes are stuck in meiosis 1 for years and years and years before potentially being ovulated and used. And this is probably the ultimate cause of evolutionary cause of menopause. Why? If indeed natural selection picks individuals who are the best reproducers, how in the world would it be advantageous for a women's reproductive system to shut down, which is essentially what happens during menopause? How could that ever be selected for if we select for maximal reproductive capabilities? Well, in the case of the human female, once you hit a certain age, the odds of producing a normal oocyte after all those years of being stuck together in meiosis 1, the odds of producing a normal oocyte become so low that the body essentially just stops trying. That's what we think is the ultimate evolutionary cause of menopause, the shutdown of the reproductive system. And so rather than have your own offspring, which are not likely to be normal, evolutionary theory predicts that the female would then shift her focus to assisting relatives in raising their children, and that becomes the best way to ensure your genetic contribution to the next generation. If you can't do it yourself, of course, you help your next closest relative. At least that's what evolutionary theory predicts. Okay, so what happens to these 200,000 oocytes once puberty begins? At this point, we enter a fairly complicated stage of oogenesis. And so what happens is when we hit puberty or adolescence, egg development can resume. As we mentioned, during puberty you get a surge in FSH. And this increase in background levels of FSH will stimulate groups of primary oocytes to begin to complete meiosis 1. We don't just stimulate one at a time, we stimulate groups of about 24. And so we call these cohorts of primary oocytes. We stimulate them to continue meiosis 1. This, notice here another, I think, slight misstatement. It says complete meiosis 1. Well, we don't actually complete meiosis 1 while we're still a primary oocyte. So what the FSH does, it stimulates the primary oocytes to continue the first meiotic division. So they kick it back into gear, basically, pull it out of stasis and start moving it along that first meiotic division again. Eventually, each oocyte, each primary oocyte, is going to complete the first meiotic division and produce 2 daughter cells. And these will be haploid daughter cells, remember, because the first meiotic division is what cuts the genetic material in half. Now, when does this division occur? It actually occurs right before ovulation. It actually occurs right before ovulation. So we don't finish that first meiotic division until literally the same day as ovulation in the 28-day cycle. And again, we'll remind you of this when we go through the 28-day cycle. Another difference between male and female is this first meiotic division is not an equal division. It's an unequal or uneven division that produces one very large cell and one very tiny cell. The large cell is the functional oocyte, which is now called a secondary oocyte. The secondary oocyte is the large daughter cell produced at the end of meiosis 1. The second cell is referred to as a polar body, and this is a very, very small cell, essentially a nucleus with a thin coating of cytoplasm around it. And this polar body is not a functional gamete. It is too small, and it ultimately just disintegrates. It erodes away. So why this uneven division? Again, another difference between male and female. Males, remember, produce the smaller motile gamete. So the male gamete's job is to swim. The female produces the larger, remember, sessile gamete, meaning not capable of independent motion. So what's happening is the male is essentially just concerned with contributing genetic material to the offspring. In order for that offspring to get a good head start, it has to get most of its cytoplasm and organelles from the mother, because the sperm cell is not giving those things to the zygote. And so in order to for a zygote, a fertilized egg, to have a chance, it has to be a fairly large cell with resources. And so the female can't afford to have an even division of the primary oocyte, because that will produce two cells that are, while bigger than polar bodies, are not big enough each to provide initial support for the zygote. And so she's forced into having this unequal division and producing only one large oocyte, secondary oocyte, and this is going to be the functional cell from the first meiotic division. So she's trying to produce one cell with as much cytoplasm as possible, and the second cell is just a castaway, really, and that's the polar body. All right, so what happens to this secondary oocyte, this cell that's just completed the first meiotic division? Well, as it turns out, the secondary oocyte is actually what's ovulated. And this is why I said earlier that technically you don't ovulate an egg cell or an ovum, because technically an egg cell or an ovum is a cell that's already completed the two meiotic divisions. A secondary oocyte that's ovulated has only completed the first meiotic division. It has not even completed both meiotic divisions yet. Another interesting observation. So the secondary oocyte, again, which is produced just before ovulation, usually the same day as ovulation, it will develop as far as metaphase 2, the second, again, the metaphase stage of the second meiotic division, and once again it arrests there. And so what is ovulated is a secondary oocyte that is stuck temporarily in metaphase 2. If that secondary oocyte is not fertilized by a sperm cell, then that's the end of this story. There is no further development, and the secondary oocyte simply dies and is sloughed off during menstruation. If, however, it is fertilized, the penetration of the secondary oocyte by the sperm cell is the trigger, the stimulus, for that secondary oocyte to then go ahead and complete the second meiotic division. So the oocyte, the secondary oocyte, doesn't even bother finishing the second meiotic division unless it's fertilized. And so essentially what happens is it holds the male nuclear or the male genetic material off to the side after fertilization, goes ahead and completes its second meiotic division, has another unequal division in the second meiotic division, producing a second polar body, which is cast off, and then a functional cell, which then will allow fusion of the male chromosomes with the female chromosomes to form a new nucleus. And of course, at that point, it is referred to as a zygote. Post-fertilization, it is referred to as a zygote. Okay, so again, the chromosomes then unite following that second fertilization. And as I said, we have the fertilized egg, the zygote. And that's the beginning of a new individual. And again, initial cell division and support is provided by the fact that the secondary oocyte was so very large because of the unequal division. Eventually, the endometrium of the uterus, once implantation takes place, will nourish the developing embryo. And of course, eventually a placenta forms as well. So here we see a visual guide to what we've been talking about. The ovarian cycle is here in blue on the left, and what we've just been talking about on the right, they're showing you what stage of follicular development we are in as this happens. So again, before birth, before the female is born, we have the production of millions of oogonia. These oogonia all will initiate the first meiotic division and become primary oocytes that are arrested in meiosis 1. By the time you reach adolescence, remember, that many of these, the vast majority of these primary oocytes have died off. And so instead of having millions, by the time you hit puberty, you have about 200,000 or so, which again is plenty for the female's lifetime supply. Where is this happening in terms of structurally in the ovary? These original oogonia and the primary oocytes that are remain all takes place in the or the primary oocytes in the embryo and fetus are all in what we call the primordial follicles, the ones lined with simple squamous epithelium. All right, during adolescence, remember, we start to stimulate cohorts of about 24 primary oocytes to continue with the meiotic division. That happens, those 24 cells that begin to kick into gear and pick up meiosis again, that happens inside what we call the primary follicle that has the cuboidal lining. Remember, the primary oocyte is not going to actually do the first meiotic division until right before ovulation. So throughout the primary follicle and the secondary follicle, we have the developing primary oocyte in those types of follicles. It's not really until we reach the vesicular or Graafian follicle that we get the completion of the first meiotic division and the formation of the secondary oocyte. Notice the polar body is just cast off and dies, so most of the resources are in the secondary oocyte. So this production of the secondary oocyte happens at the center of the Graafian follicle, usually within hours before ovulation. Okay, so during ovulation, what is actually ovulated is the secondary oocyte that just completed the first meiotic division. Again, as we said, if it's not fertilized, what happens? Nothing happens. The secondary oocyte does not complete the second meiotic division. It simply dies. If, however, it is fertilized, then what will happen is the male genetic material will be kept off to the side, and the secondary oocyte will go through its second meiotic division, producing a second polar body, which dies, and a functional zygote, which then allows the sperm chromosomes to fuse with the egg cell chromosomes, producing a new diploid nucleus, and that is the zygote. And then, of course, cell division then ensues, resulting in the embryo. And of course, while all this is happening and implanting on the wall of the wall of the uterus itself is made receptive by hormones coming from the corpus luteum, which is, of course, what as we said, the Graafian follicle or vesicular follicle turns into following ovulation. So notice the timing of this. We get the corpus luteum producing its hormones like progesterone just when the zygote is ready to implant on the wall of the endometrium. And here we see again a view of the histological view of the ovary. Most of the follicles near the surface are these primordial follicles just waiting to get turned on. Again, even though you have 200,000, only about 24 get turned on every month or so. So they have to wait their turn essentially, and most of them, of course, will never get turned on. Of the ones that do get turned on, they start to develop into primary follicles, and then secondary follicles, and then finally they will become the Graafian follicles. Now, we said we initiate development of about 24 as a cohort. Of those 24, generally speaking, only one makes it as far as becoming a mature or vesicular follicle. So of the 24, generally speaking, only one makes it all the way to become a Graafian. The others degenerate along the way. It is possible that 2 or more might become Graafian follicles during the same cycle. And of course, then you would have multiple ovulation events, and that is what leads to conventional twins, fraternal twins or triplets or things like that. But really, the human reproductive system is only designed to allow one vesicular follicle to ovulate and to house one offspring at a time. Though obviously it's possible to have more than one at a time. All right, so I've just described the correlation of oogenesis to the condition of the follicles. In these next few slides, we're going to talk about these, again, follicles as they develop from primordial follicles to Graphian or vesicular follicles. There's a little bit of a twist here that's a little bit surprising. Up till now, it's possible that you may be thinking that it takes about 28 days for a primordial follicle to become a vesicular follicle, because we keep harping on the 28-day cycle. But actually, it takes a lot longer than that. So even though we ovulate every month, it doesn't mean that the secondary oocyte that's ovulated each month just started or just came from a primordial follicle 28 days earlier. It's actually much longer than that. So again, we're not totally responsible for this, and we don't really have these details in our list of terms, but it's so interesting that I just want to talk about it briefly. So again, remember, primordial follicles, we have about 200,000 of them, and they contain the primary oocytes that are arrested in meiosis, and they're surrounded by a single layer of squamous cells again. They're mostly found in the cortex of the ovary, and again, because we have 200,000 of them and only 24 or so get activated every 28 days or so, some of them have to wait as much as 50 years or more in some rare cases before they actually develop any further. The vast majority of them never develop beyond just being arrested in meiosis. As we said, there's a monthly recruitment. Every 28 days we recruit about 24 follicles to begin their development. But to make it from, and here's the twist, to make it from a primordial follicle all the way to a vesicular follicle, as I mentioned, hinted at earlier, doesn't take just 28 days. It takes 290 days to go from primordial follicle to mature vesicular follicle. 290 days is just under 10 months. So it's really quite a long cycle to go from primordial follicle to Graphian follicle. So how do we end up ovulating every month? Well, every month we're ovulating from a follicle that began its cycle 290 days earlier. So as long as we keep initiating cohorts every 28 days, eventually we end up ovulating 28 days, every 28 days. But what we ovulate is a secondary oocyte that's just completed a 290-day path to becoming a secondary oocyte in a Graphian follicle. And again, of the cohort of 24 that initiates this 290-day cycle or path, only about one usually makes it. The other 23, as I said, die off, except in the case of fraternal twins or fraternal triplets, where more than one makes it the whole way. So about 140 days into this 290-day path, in other words, about halfway into it, the 24 primordial follicles will become primary follicles. These primary follicles contain the oocyte again. Now here again is a typo in the presentation. The PowerPoint says that the primary follicles contain the secondary oocyte. Well, that's not true. Remember, the secondary oocyte is the cell that is derived from the first meiotic division, and that doesn't happen until right before ovulation. So it's impossible for this to be a secondary oocyte. So the primary follicles still contain the primary oocytes that have not completed meiosis yet. They have been, this cohort of 24 has been jogged into completing meiosis, but they haven't completed meiosis yet. It takes a long time for them to complete that first meiotic division. Okay, we know primary follicles then turn into secondary follicles. Here we have another typo because it says, the PowerPoint originally says that the secondary follicles appear about 140 days into the cycle. Well, that's impossible because the primary follicles appear at 140 days into the cycle. So it has to be later than that. And in reality, it's 170 days into the cycle. So what this means is that The primordial follicles occupy about the first half of this 290-day path. And then we get primary follicles for about 30 days. And then 30 days after the first primary follicle formation, then they convert to secondary follicles. And in these secondary follicles, the oocytes continue to enlarge, getting ready for that first meiotic division. And again, we have the very characteristic structures of the secondary follicle that we've already described, the thicker zona granulosa, etc. This slide, again, another sort of mistake. It essentially repeats the previous slide. Just it provides a little more detail about the structure, specifically called the theca folliculi, which is more detail than we're covering in this presentation. All right, so when do we start to get to Graphian or mature follicles? And again, your book is referring to these as tertiary follicles, but I'm gonna consider this to be the beginning of the mature follicles. Well, they occur about 60 days before ovulation. And so remember, we were at day 170, now we're at day 230. So the secondary follicle lasts about 60 days. And then the vesicular or mature follicle is going to occupy about the final 60 days of this 290-day cycle. And so once again, what happens here is the antrum forms and begins to fill with fluid. The egg starts to project. The oocyte, which again is still a primary oocyte, projects into the antrum, and eventually we end up with the mature follicle with the C-shaped antrum and the oocyte projecting into the center of it. All right, this final appearance of the mature follicle with the C-shaped antrum and so on, when it looks like a typical Graphian or vesicular follicle, is about day 270 in the cycle. About 20 days before ovulation is when it starts to have that characteristic C-shaped antrum. And again, only one follicle of the 24 that started this cycle 270 days earlier, only one of them generally reaches this point. By this time, the rest have usually degenerated. At this point, it begins to capture and hold on to FSH. That's coming from the pituitary gland, the anterior pituitary. And as we'll see in a few slides, FSH and LH are going to have play an important role in determining when exactly this Graphian follicle is going to ovulate. As we said, just before ovulation, meiosis 1 is completed, and so the secondary oocyte is sitting there ready to go in the Graphian or vesicular follicle just before ovulation. Okay, so that's the story of oogenesis and how it correlates with the follicles, the different follicles that we see. Now, your authors like to talk about the ovarian cycle in 3 phases. This is a 28-day phase. So we're not talking about the follicular development. We're just talking about a 28-day cycle now of what we observe happening in the ovary. And your book breaks this up into follicular phase, ovulation, and luteal phase. We don't have these terms in our list of terms, but I want to talk about them because it does help us understand the menstrual cycle, which is what we're going to finish up with today. So the follicular phase, again, now we're talking about a 28-day cycle. The follicular phase represents the first half of this cycle, day 1 through 14. And what happens during the follicular phase is essentially that final development of the tertiary follicle into the full-fledged Graphian or vesicular follicle. In other words, the final follicular development, getting the follicle, the one chosen dominant follicle, ready for ovulation. Again, FSH, follicle stimulating hormone, is the important hormone that stimulates this dominant follicle to grow into this final pre-ovulatory phase. And that's, of course, where the name follicle stimulating hormone comes from, because it is the hormone that stimulates this final development at the end of the 290-day cycle for that dominant follicle to get ready for ovulation. As the dominant follicle develops, it becomes increasingly sensitive to FSH and LH, almost a positive feedback effect. And it also becomes sensitive to estradiol, which is the most common form of estrogen. Where's this estrogen coming from? Well, it's coming from the follicle itself. The estrogen comes from this Graafian or vesicular follicle as it develops. And as it develops, the levels of estrogen that it produces gradually increase. So it starts, when it's a tertiary follicle, it starts at fairly low levels, and then as we approach day 14 of the cycle, the estrogen is continually increasing as we go through this follicular phase. Ovulation is a very short-term event in the cycle, in the ovarian cycle. It typically happens around day 14, though of course there's lots of variation associated with this. It can be a few days earlier or a few days later. It's generally not going to be day 2 or day 27 or 26, but it is going to cluster usually around day 14, including a few days before and a few days after. This is important if you're using a rhythm method of birth control or a rhythm method of trying to get pregnant. Of course, you want to try to fertilize the egg somewhere around day 14 in this cycle. At any rate, ovulation, of course, is, as we said, a rupture of the vesicular follicle and the release of the secondary oocyte and some of its surrounding tissues. The trigger for ovulation is a surge, a dramatic surge in luteinizing hormone. There's a little bit of a surge in FSH as well, but the most dramatic surge is in a hormone called luteinizing hormone. And what stimulates that, as we'll see, we have a graph that shows all this coming up. What causes all that is the increase in estrogen. Remember, as we approach ovulation, the vesicular follicle continues to produce more and more and more estradiol or estrogen. And finally, the estrogen levels get high enough that triggers a surge, a spike of luteinizing hormone and FSH. And that spike is the trigger for ovulation. So what does this spike of LH do? It triggers the completion of the first meiotic division. Again, this is what causes a secondary oocyte to be produced right before ovulation. It causes a rapid increase in the formation of follicular fluid, which causes the antrum to swell inside the vesicular follicle and eventually burst. It stimulates macrophages and other white blood cells to secrete enzymes to weaken the follicular wall, also encouraging it to burst open. And just before ovulation, you see a little protrusion of the antrum and its fluid through a little opening on the surface. This is referred to as the stigma. Looks like a little nipple protruding from the vesicular follicle just before ovulation. So in terms of the hormonal regulation, this is a first look at what's happening. Remember, the maturing follicle, as it's approaching ovulation, is secreting estrogen. And that initially, interestingly enough, the lower levels of estrogen tend to have an inhibiting effect on FSH and LH. But just before ovulation, we get high enough estrogen levels that its effect reverses. And the higher levels of estrogen actually have an excitatory effect on the hypothalamus and the pituitary. It stimulates the hypothalamus, of course, to release its gonadotropin-releasing hormone, but the estrogen also directly affects the anterior pituitary, and so through both these mechanisms, we end up with an increase in the release of FSH and LH. The more FSH that is released, of course, that stimulates further follicular development, so you sort of have this positive feedback loop, but it's the final spike and surge in LH and FSH that triggers the events leading to ovulation. Again, the ovulatory phase or the ovulation phase is very short-lived. It only takes 2 or 3 minutes to complete the ovulation phase. The stigma, the little nipple-like structure, might seep fluid about for a couple of minutes before ovulation, but the actual explosion itself, the rupturing, only takes about 2 or 3 minutes. And again, that secondary oocyte that's released normally should be swept up by a current created by the uterine tube and start flowing or being carried towards the uterus. Here we see a photograph of the stigma, the nipple-like protrusion containing the secondary oocyte protruding from the wall of the ovary just before ovulation. Again, the uterine tube is what normally should catch the oocyte and begin to carry that egg towards the uterus. Now, again, if you're using some kind of rhythm method to try and get pregnant, it's important to know when ovulation has occurred. So, of course, you want to try around day 14. You also want to be sensitive to various signs of ovulation. The cervical mucus changes and becomes thinner. The body temperature rises slightly during ovulation. And of course, body temperature fluctuates quite a bit anyway, so it's best to be monitoring temperature over several days to be able to pick up on an abnormal fluctuation. And in some cases, women can actually feel little sensations of pain during ovulation. The best time, of course, for conception is within 24 hours following these changes, because again, you want fertilization to occur while the secondary oocyte is in the uterine tube. If you wait too long, then it's going to be, again, too late. All right, just to complete the ovarian cycle, the third phase, or the last half of the 28-day cycle from the ovaries perspective, is called the luteal phase. And of course, this is when we get the formation of the corpus luteum after ovulation. Now, in most cases, pregnancy won't occur, in most cases, throughout a woman's life. And so what happens if pregnancy doesn't occur is that, again, the ovulated follicle, first of all, bleeds and then repairs itself, and it gets a dense bed of capillaries growing within it as a result of this repair. And the development of glandular tissue, the granulosa cells become glandular tissue, and it essentially converts to a corpus luteum. And as we said, that's named for the yellow lipid, fat tissue that accumulates inside. And this corpus luteum is going to produce its own set of hormones. And again, this transformation into the corpus luteum is once again stimulated by luteinizing hormones. So that spike of LH that occurred to promote ovulation also stimulates conversion to the corpus luteum. And of course, this is where the name luteinizing hormone comes from, because it stimulates conversion to the corpus luteum. So that's the luteinizing part. The most noticeable hormonal change that results from formation of the corpus luteum is the production of the hormone progesterone. And so we see a dramatic increase in progesterone production during the luteal phase, because it is being secreted by the corpus luteum. Progesterone, of course, is critically important in getting the endometrium of the uterus ready for implantation and pregnancy. At this point, LH and FSH begin to decline because of negative feedback from progesterone and estrogen again. Remember, at lower levels, estrogen has an inhibitory effect on LH and FSH, and progesterone certainly has an inhibitory effect on LH and FSH. And so that happens over the next few days. Remember, there's also inhibin being produced by the ovaries from various follicles and from the corpus luteum, and that also has a negative feedback. And so our levels of FSH and LH are pretty low during this luteal phase while we're producing progesterone. Now, this lasts until about day 22 in the cycle, in the 28-day cycle. So for about 8 days after ovulation, we have this high level of progesterone. Now, again, in the cases where pregnancy doesn't ensue, then what happens is the corpus luteum, starting at about day 22, starts to degrade. It goes through what's called involution, and the glandular tissue degrades, and you essentially, by day 26, what you're left with is just a small patch of scar tissue. And again, remember, that's called the corpus albicans. And so from day 22 to day 26, you've got, of course, a reduction in the progesterone, especially from the LH, as the corpus luteum degrades. And this reduction in progesterone is what then allows FSH levels to rise back up again. And as FSH levels rise back up again, we set the stages for a new cohort of follicles to start their development. And we start a new 28-day cycle over again. And again, it's not a true 28-day cycle. We start a new 290-day path. But remember, there's already a set of follicles ready to go for the next 28-day cycle. Now, this is a diagram that I took from another book because I think it shows a little bit better than our previous diagram what the various hormonal interactions are going on. And we do have a set of terms in our list of terms under endocrinology that we want to refer to here. And so we begin again with the anterior pituitary at the beginning of the 28-day cycle producing levels of FSH and LH. This FSH and LH are going to stimulate the follicles, again, the ones that are especially near the end of their 290-day cycle. It's going to stimulate them through various mechanisms to produce estrogen, specifically estradiol. It's also going to stimulate them to produce the hormone inhibin. All right, and so as these follicles develop, as these secondary follicles develop, they're going to produce more and more estrogen and inhibin, and the estrogen that's being produced is going to feed back onto the follicle itself and help promote its maturation, eventually into a mature vesicular follicle. Now, initially, as we said, the relatively low levels of estrogen that we start with at the beginning of the cycle, plus the inhibin that's being produced, will have an inhibitory effect on the hypothalamus and the pituitary gland. And so levels of FSH and LH are going to drop. So we've initiated the dominant follicle to complete its development by initial high levels of FSH and LH. But now, as we progress into the 28-day cycle, we have an inhibitory effect on FSH and LH. And again, that's through early or lower levels of estrogens and inhibin. As the secondary follicle develops into a tertiary follicle and eventually into a mature follicle over the course of the 28-day cycle, which of course is near the end of the 290-day path for this particular follicle, it secretes more and more and more estrogen, and again in a positive feedback loop. And so eventually the estrogen levels rise to such a high level that its effect reverses. So again, remember, initially low levels of estrogen have an inhibitory effect, but towards the middle of the 28-day cycle, estrogen levels have risen so high that now they have a positive effect on the hypothalamus, causing it to release its gonadotropin-releasing hormone, which stimulates the anterior pituitary to release its surge of luteinizing hormone, and to a lesser extent a surge in FSH, but it's really the luteinizing hormone surge that's important here, because what that leads to, remember, is ovulation. The surge in LH is what stimulates the ovulation event. And following ovulation, of course, the LH surge also stimulates the ruptured vesicular follicle to convert into a corpus luteum. Again, this corpus luteum for about 8 days will produce high levels of progesterone. Those high levels of progesterone are going to inhibit FSH and LH and prepare the endometrium for implantation in pregnancy. If no pregnancy ensues, then remember, after about 8 days, the corpus luteum begins to degenerate into a corpus albicans. And in the absence of progesterone, we start to see LH and FSH production come back up. And when that happens, we end up back where we started. Levels of FSH and LH come back up, and we start the cycle all over again for another 28-day cycle. Now, if pregnancy does ensue, we haven't talked about what happens if pregnancy does ensue. If pregnancy does ensue, then we have a variety of hormones that are produced by the implanted zygote as it develops. One of which is very well known, human chorionic gonadotropin. And these hormones that are coming from the implanted zygote are going to set up signals that will maintain the corpus luteum for the full 9 months of pregnancy. So as long as the body knows that it's pregnant, and HCG, human chorionic gonadotropin, is one of the hormones we look for in pregnancy tests, for instance. The body knows it's pregnant from those signals, and the corpus luteum will be maintained for the full duration of pregnancy. So it will persist for 9 months in the case of pregnancy. And of course, the progesterone that it makes will continue to support the pregnancy, and the progesterone that it makes will continue to inhibit FSH and LH. And so this is why you don't have a new 28-day cycle begin once you become pregnant, and you don't have any ovulation occurring during pregnancy. It's because progesterone levels remain high throughout the pregnancy, and remember, that inhibits LH and FSH. So we don't start this cycle here again until after pregnancy is over. And there's some evidence that breastfeeding also through oxytocin will create some signals that will inhibit FSH and LH as well, though there have certainly been cases where breastfeeding mothers have gotten pregnant fairly quickly. So it's not a foolproof form of birth control. All right. So how about the menstrual cycle? I apologize for the typo here. It should be menstrual cycle. The menstrual cycle is, again, this 28-day cycle, but now we look at it from the perspective of not what's happening in the ovary, but what's happening with the endometrium specifically. So again, it averages 28 days, but there's variation. It can range to be as low as 20 or as high as 45. In some cases, the cycle is skipped completely. That does happen, of course, as well. It is primarily regulated by the same hormones that we've been talking about, gonadotropin-releasing hormones from the hypothalamus, FSH and LH from the pituitary, and estrogen, progesterone, etc. from the ovaries. And so essentially what happens in the menstrual or uterine cycle is we begin again with a 2-week phase. According to your book, there's 2 portions to the menstrual cycle, the follicular phase and the secretory phase. Unfortunately, in our list of terms, we break it up into 3 phases. So basically, we take what your book calls the follicular phase and we break it up a little bit differently. And so again, this is one of the differences between how our list of terms looks at it and how we look at it. I'm going to try as best I can to stick to the list of terms. And so the cycle begins, the 28-day cycle begins with the first day of menstruation. So day 1 would be the first day of your period, of the menstrual period. This is what we call the menstrual phase, and it lasts generally from 3 to 5 days. All right, so important to understand that the cycle, the 28-day cycle, begins with the beginning of menstruation, not the end of menstruation. And of course, during menstruation, what's happening is that the endometrium is sloughing off because pregnancy did not result, and we're getting rid of that functional layer of the endometrium, and it will again over the next 23 days or so be replaced with a new functional layer coming from the basal layer. And so the first, again, 3 to 5 days is the menstrual phase. That's then followed by the proliferative phase. And the proliferative phase lasts up to about day 14. So let's call it 5 days for the menstrual phase and then another 9 days for the proliferative phase. During the proliferative phase, the uterus is replacing that lost endometrium and building up a new functional layer of endometrium. Again, as we've said repeatedly, around day 14, ovulation occurs. And at that point, after ovulation, remember, the ovary converts to become a corpus luteum. And so the remainder of the 28-day cycle, the remaining 14 days in our list of terms, we refer to this as the secretory phase. And during this secretory phase, the corpus luteum secretes, again, progesterone, and that stimulates the endometrium to continue its thickening, again, anticipating pregnancy. If pregnancy ensues, of course, then for 9 months, you're going to have essentially this secretory phase where the corpus luteum is active and maintains pregnancy. If pregnancy does not occur, remember, the corpus luteum begins to break down, and as the corpus luteum breaks down and progesterone levels fall, the endometrium will begin to break down. And so at the end of this secretory phase, in the last 2 phases or so, the endometrium begins to break down, and at that point, at the end of 28 days, we go back to where we start with menstruation, and we start a new 28-day cycle with the next menstrual phase. Here we see a nice graphical representation of what's happening. I wouldn't pay too much attention to exactly what's happening down here. It's a little bit misleading because, again, during a 28-day cycle, you do not move from primary follicle to ovulation. It takes, remember, 290 days to go from a primordial follicle to ovulation. So this is a little bit misleading, but what does happen during this 28-day cycle is that a tertiary follicle that has been produced after about 270 days or so is ready to finish its completion of its development to ovulation. Other follicles get stimulated along the way. Of course, they want to continue their 290-day cycle, so they respond to the FSH and the LH by continuing their 290-day development. But this is misleading if you think that it means that we go from a primary to ovulation in 14 days. That's not the case. But what we mainly want to notice here is that FSH and LH levels are relatively high at the beginning of the 28-day cycle. And then FSH especially is inhibited by low levels of estrogen. Then right before ovulation, remember, the increasing levels of estrogen cause a spike, especially in LH. Notice a little spike in FSH as well, but primarily a spike in LH that we talked about. And that's the stimulus for ovulation and then conversion of the vesicular follicle into the corpus luteum. The corpus luteum is going to produce progesterone, which is going to keep levels of FSH and LH a little bit low until we get the degradation of the corpus luteum into a corpus albicans. And once we get the corpus albicans, we lose the progesterone, and so FSH starts to come back up. And that increase in FSH is going to stimulate a new cohort of, say, 24 primordial follicles to initiate their 290-day path of development. Here we see it from a different perspective, and in this case we're looking at what happens to the lining of the uterus during the 28-day cycle, and we're following the 2 other hormones now, estrogen and progesterone. So notice that Initially, again, the first 5 days or so are the menstrual phase, and we get a loss of the functional layer of the endometrium. So at the end of 5 days or so, the endometrium is at its thinnest, consisting of just the basal layer and a little bit of new developing functional layer. Over the course of the proliferative phase, of course, we start to build up the endometrium again. During this time, we have relatively low levels of estrogen, which are inhibiting primarily FSH, also inhibiting LH a little bit. But notice, as we approach ovulation, as that Graafian follicle develops, we get a surge in estrogen. And that surge in estrogen is going to be what triggers the LH and FSH spike. The LH and FSH spike will cause, again, ovulation and conversion of the Graafian or vesicular follicle into the corpus luteum. And so following ovulation, we see a dramatic rise in progesterone. Again, remember progesterone being produced by the corpus luteum. We also see that estrogen remains fairly high during this time. This high level of progesterone specifically inhibits FSH and LH and causes them to stay relatively low through the rest of this 28-day cycle. And so for almost the duration of the 28-day cycle, we continue to have thickening of the endometrium, again, readying itself for pregnancy or supporting pregnancy if pregnancy has ensued. This is called the secretory phase. But notice in the last couple of days of the secretory phase, the corpus luteum is now degenerating into a corpus albicans, so levels of progesterone drop off dramatically. And that drop in progesterone causes the endometrium to start to degrade in anticipation of menstruation. And when we get to the end of the 28-day cycle, of course, that endometrium is going to slough off and we begin the next menstrual phase, and the whole cycle repeats itself. And so that's the endocrinology associated with the female sexual cycle. And here we see just a view of the changes of the endometrium during each phase that we just described. The last slide for this presentation, just a nod to endometriosis. It's not on our list of terms, but it's something you've heard of, no doubt. Endometriosis simply means the growth of the endometrial type tissue in some place other than the uterus. It can happen on the peritoneal membrane. The roof of the pelvic cavity, remember, is the parietal peritoneum. So it can grow on the undersurface of that. It can grow on the surface of the uterus, the outer surface of the uterus. It can grow on the surface of the ovary, the uterine tubes. Generally, it's painful as this endometrial tissue grows and develops in these abnormal areas. Sometimes, if it happens on the ovary, it can cause ovarian damage, resulting in infertility. Oftentimes, it seems to result from retrograde menstruation. So, again, during menstruation, of course, the endometrium is supposed to slough out through the vaginal canal. But it's possible that there might be a backward flow of menstrual fluid through the uterine tubes and then out into the pelvic cavity. And so that's something that has to be watched. It's not too common, but it's not rare either. 6 to 10% of women will experience endometriosis sometime during their lives. Okay, so that's it for the female reproductive system. There will be a short second video. Once again, we have 2 models for me to show you these structures. And so I look forward to seeing you in a few minutes on that model video.